This is the Pharmacology and Toxicology Departmental Seminar, and the first Thursday of the month is the Environmental Health Science Series, hosted by the Center for Integrative Environmental Health Science. And we're very pleased to have an external speaker here in person. And to introduce our speaker, Dr. Greg Barnes, who's the leader of the Neurodevelopmental Toxicology Research Interest Group. Let's have that on.
So I'm going to stand up here so that but you can't hardly see me. So for the people that I need a little step. All right, so it's such a pleasure to welcome Dr. Deborah Cory-Slechta, who is a professor of environmental science, environmental medicine, pediatrics, and public health at the University of Rochester Medical School. And she is the former chair of the Department of Environmental Sciences, and she is the PI on the NIEHS Core Center grant there at Rochester. She has served previously as the Dean of Research at the University of Rochester's Medical School and Director of the Environmental and Occupational Health Sciences Institute at Rutgers University. Her research includes both animal models and human studies focused largely on the consequences of developmental exposures to environmental chemicals on brain development and behavior. This work has examined the effects of developmental exposures to metals, pesticides, and air pollutants in animal models and human cohorts. These efforts have resulted in over 200 wonderful peer-reviewed publications. Dr. Cory-Slechta has served on numerous advisory panels of the NIH, the FDA, the EPA, the National Academy of Sciences, the Institute of Medicine, the Agency for Toxic Substances and Disease Registry, and she serves on numerous editorial boards, including the Environmental Health Perspectives, Neurotoxicology, Toxicological Sciences, Toxicology and Applied Pharmacology, and Neurotoxicology and Teratology. She has also served on the U.S. EPA Science Advisory Board and the Board of Scientific Counselors for the CDC. In 2017, she was the recipient of the Distinguished Neurotoxicologist Award from the Neurotoxicology Specialty Section of the Society of Toxicology. In 2021, she was awarded the Distinguished Toxicology Scholar Award from the Society for Toxicology. So would you join me in a warm welcome of Dr. Cory-Slechta?
If that happens again, I'm just going to click back on PowerPoint.
Okay. Well, I don't know if anybody can see me. I have the same problem, but first of all, I just want to thank you so much. It's really my first trip back out too in the last 15 months. So it's been really an interesting journey, and I'm very pleased to be here. It's great to see people sitting in a room and not just on a Zoom meeting. So again, I want to thank you very much. I'm going to talk today about ultrafine particle air pollution and its impact with respect to neurodevelopmental disorders. And just as a preface, I will say that I was pulled into this a little bit kind of against my better judgment at the time. I had been working for a long time on lead exposure, and I thought, you know, lead kills kids. How bad can air pollution be? Because one of my colleagues said, gee, I just did this study. I exposed animals. I want you to look at the brains. And I thought, oh, okay, all right. So we looked at the brains, and boy, that was the start of a new journey. So let me start, for those of you who aren't familiar with air pollution. It's really all about particle size. And what you're looking at here On this side is a human hair, so that's 50 to 70 microns in diameter. And then we talk about 3 different classes of particle sizes, the largest being what's called PM10, coarse particles. Those are 10 microns in diameter or less, and I think you can see them relative to the size of the human hair. There's then the fine particulate class. These are 2.5 microns in diameter or less that you can see in the blue. And the ultrafine particle component, these are nanoparticles, less than 100 nanometers in diameter, and you can see many of those superimposed on a fine particle. Ultrafine particles are considered the most reactive component of air pollution for reasons that hopefully you'll see as we go along. Partly, it's all of the chemical contamination that is adsorbed onto that ultrafine particle. And here what you can see is the elemental carbon core of an ultrafine particle, and to that is adsorbed organic carbon species, trace metals, redox active metals and elements. There are secondary reactions that go on, chemical reactions in air pollution, so we get secondary sulfates and nitrates. This is probably the most complex mixture that you can have in terms of toxicology. And again, this ultrafine particle, nanoparticles, being the most reactive. This is really a lifelong exposure. One of the things that happens, let's say during pregnancy, is that the mother is breathing in air in a couple of ways, which you'll see again in a minute. Those particles can get directly, they can contact the placenta directly and cause damage that way. But the particles, in particular the ultrafine particles, can also cross the placenta and cause inflammation and also get directly into the fetal bloodstream. So even in utero, this is already a wide open, if you will, kind of exposure. What happens after you're born is that this exposure continues, but now it includes other routes of exposure. So you breathe in the air. Those ultrafine particles are so small that they get into the deepest parts of the lung. They basically can bypass macrophages that are there that would normally pick up this kind of debris and get directly into the bloodstream. In addition to that, you are breathing these particles up your nose. And we now know from many different studies that those particles can directly access the brain via being taken up on the olfactory axons, go directly into the brain. So you're bypassing all of the regulatory mechanisms that would normally keep contaminants out of the brain. This, again, is a lifelong process. In addition to that, you can get, these particles will then travel along not only the olfactory nerve, but the trigeminal nerve and spread throughout the brain. But there's also now an indication that the vagal nerve, which is a peripheral nerve that connects virtually every organ and the brain, can also carry these particles and their contaminants and go directly into the brain stem. So lots of routes of exposure. This is a lifelong exposure. So there has been a growing body of evidence for a role of air pollution effects in neurodevelopmental disorders. And here I'm referring to the epidemiological studies that have come out to date. There's probably something like 20 studies right now that have associated air pollution with autism. There are studies associating it with schizophrenia, attention deficit disorder, a decline in the cognitive trajectory that children would normally show across the years, and something called interhemispheric disconnectivity, which I'll talk about in a minute. So there's some intriguing correspondences that you can find just by looking at maps. On the left here, I'm showing you data of, this is PM 2.5 air pollution in the US, and you can see that where the highest areas are clearly here in the Midwest, up and down the East Coast and the West Coast. What's interesting, clearly not necessarily causal or even association, is if I look at a very recent map of autism prevalence in the United States, and I think you can see in many ways the overlap to what we're talking about with something like PM 2.5. Again, it may be nothing more than simply a just casual, nothing that's causal, but interesting. Here's another one for ADHD prevalence with respect to air pollution. And again, you can see where some of the highest prevalence rates for ADHD are in relation to air pollution. So there's some very intriguing correspondences that I hope will get followed up with geographical mapping technologies and some of these kind of data. So you're probably asking yourself at this point, well, we know that air has been getting cleaner, right? Because EPA has been, certainly in the US, regulating levels of PM10 and PM2.5, but at the same time, the rates of neurodevelopmental disorders seem to just continue to be increasing. Maybe some of them are slowing down a little, but they've been increasing. So how could air pollution then play a role in something like neurodevelopmental disorders? Well, yes, the levels of PM10 and PM2.5 have decreased because EPA does regulate them. But let's remember that the most reactive component of air pollution is the ultrafine particle component that is not regulated by EPA. And by taking out the PM2.5 and PM10, there's no agglomerate. The ultrafine particles can't really agglomerate on PM2.5 and PM10, so they can stay in the air longer, travel further. So it's also interesting with respect to that people have been looking at correlations between the levels of ultrafine particles and PM2.5, and you don't find correlations anymore. So there's a lot of ultrafine particle components, even with a decrease in PM2.5 and PM10. So something I want us to remember as we go through this, we think about these neurodevelopmental disorders usually in respect to the unique characteristics that they show, because each of them does have unique characteristics. But at the same time, they share multiple other features, whether it's the fact that most of the early neurodevelopmental disorders are male biased. All of them that I'm aware of show what's called ventriculomegaly. This is in the large lateral ventricle. Other features, differences, changes in myelination, etc. They can show similar kinds of behavioral deficits as well, which means that if air pollution does play a role in these, it could be a much broader risk factor than we think about. Instead of just being autism, it may be a much broader risk factor. So what I'm going to talk about today are studies that we undertook looking at this in a mouse model. And I'm going to start by talking about exposures that occurred in the early postnatal period. And this is a diagram, it's a schematic of our exposure system. We actually pull air in from outside. So this is ambient exposure. Once it's pulled in, it's separated by particle size. We then concentrate the ultrafine particle component and deliver that in whole body chambers to mice. And we have adjacent HEPA filtered air for the control animals. So this is an ambient, concentrated ambient ultrafine particle exposure, which you'll see sometimes referred to here as CAPS. And this, we started with the early postnatal period, which I'm going to talk about first. So those were exposures that occurred from postnatal days 4 to 7 and 10 to 13. They went on for 4 hours a day. They would typically start at 7 or 8 in the morning to catch all that traffic that was outside the road. And why do we pick these? Because this species of mice and most species of mice are altricial. So this is actually equivalent to the human third trimester in terms of brain development. And I'll show you that again in a minute. So this is human third trimester equivalent in a mouse. And after that, we collect tissue, do behavioral analysis, et cetera. And this is an example of that. This shows you the increase in neurons and non-neuronal cells in the brain of mice during this early postnatal period. You can see there's just marked increases in brain development going on during this period of time. There are other species of mice. One's called a spiny mouse that actually has a 39-day gestation period, so it goes through third trimester in utero, which would be very interesting comparatively speaking. Okay. So this slide shows you the development of the lateral ventricle in rodents. So the ventricular system in your brain is how the cerebrospinal fluid flows. And you can see here in a normal mouse at 4 days of age, the ventricles are pretty wide open. On top of those is a structure called the corpus callosum. It's the largest white matter tract in your brain, so it's myelinated axons, and that really holds the two hemispheres together. It literally runs from the front to the back of your brain, largest tract we have. But you can see by postnatal day 14, the ventricles have basically been compressed as the rest of that infrastructure, neuronal and glial cells, has developed over the course of that 10 days. This was the first data I got back after looking at brains of animals that have been exposed postnatally to air pollution. And on the top, this is at postnatal day 14, so it's 24 hours after this exposure is ended. You can see the brains. These are coronal sections of a HEPA air exposed male. And you can see just a little bit here the size of the lateral ventricle. These are male brains from mice that were exposed to CAPS at postnatal day 14, and I think you can see very dramatically enlarged lateral ventricle. This is damage that's, for the most part, so severe that there's little chance of significant repair. At the same time, you can see that this corpus callosum, which should be a dark brown kind of line that looks like that, almost looks like Swiss cheese and certainly doesn't seem to cross the midline of the brain, so not holding those two together. We've seen this now down at exposure concentrations as low as 44 micrograms per cubic meter, which is the lowest exposure concentration we've had to date in our studies. And at first, when I saw these, I didn't believe it, and I said to the student, you know, this could be an artifact. Did you stretch it over? What happened? Did you stretch it? Well, he said, first of all, we only see it in the males. And I thought, well, okay, that's pretty weird because they're all in the same exposure chamber. Why would just the males have it? And I said, okay, well, we have brains from other animals that we exposed that we put in the freezer. They were 270 days of age. Go get the brains, section them, look at them. If we still see this, I'll believe it. Sure enough, at 270 days of age, you can see that the lateral ventricle is still very enlarged in these male brains. At the same time that, and you saw evidence for this before, the size of the corpus callosum is shrunk, it's reduced in size, and it should consist of all myelinated tissue connecting the two hemispheres. Well, you can see in a control brain that there's about 90% myelination, this orange staining in the corpus callosum. But after this CAPS exposure postnatally, we're down to about 15% myelination. And you can see that size of the corpus callosum that's reduced here in the males as well. This is a mistake here, this asterisk. We did not see this effect in females. It was only in males. Oops, I want to go. Okay, and it actually follows the trajectory of development that's ongoing at the time of exposure. So these are different areas of the corpus callosum, the truncus, the medial external capsule, and the lateral external capsule. These were the areas here under development at the time of exposure. This happens a little bit later, and you can see that it was spared. Interestingly, again, no effects in females at postnatal day 14. We do see some repair in terms of white matter staining as males get older. What's interesting is that suddenly at postnatal day 270, it looks like we're starting to see some latent effects in females, and I'll say something more about that at the very end. The other thing that we've found in the males, again, exclusively is cell death. This is in an area called the nucleus accumbens. It's part of one of the brain's dopamine systems. It's very important to executive/cognitive function. This is silver staining that you can see here that we don't see in controls. There's an increase in serum oxidized glutathione levels, so clearly an oxidative stress kind of event going on, which again, we don't see in females with this exposure. We also see an excitatory inhibitory imbalance here, which is basically the ratio of glutamatergic to GABAergic function. It's been a studied component of things like autism and even schizophrenia over the years. You can see that effect in males, and it's still evident. Oh, let's see here. I got to go back and it's still seen in males even at postnatal day 60. In fact, by that time, it seems to have grown a little bit. Whereas it looked like there were some trends in the females, but this was never statistically significant. So one of the other things we wanted to do was look at some behavioral changes, potential behavioral changes in these animals. And one of the behavioral features of many neurodevelopmental disorders is kind of impulsive behavior, impulsivity. And so we developed a behavioral baseline specifically to look at that in mice. And basically they're in an operant chamber, a Skinner box, which hopefully most of you know about, where they had to press a lever 50 times. And at the 50th, when they made the 50th response, they get a food reward. So they're food motivated by caloric restriction. And then they don't have to do anything. They don't do anything. They just keep getting free reward deliveries. But the trick here is that the time between each reinforcer delivery just keeps getting longer and longer. So you have to patiently wait. And you can see what that looks like here. Now they have the option to make a response and start this whole thing over again. But again, you got to make 50 responses to get back there. These are what we call cumulative records of their performance. This is a cumulative record from an air control on the top. You can see this guy made his 50 responses. He starts picking up all kinds of free rewards, starts over again, picks up a couple, but he has very long periods of just waiting behavior where he's picking up all these reinforcers. I think it's quite clear in the bottom cumulative record of the CAPS-exposed male that there's very little waiting behavior that goes on whatsoever in these particular animals. If we quantify that, the mean time that they wait, which is here, is significantly reduced relative to control. If I re-expose these guys as adults for another 4 days, the effect is even a little bit worse. And the number of times they actually reset that 50 response requirement is obviously much, much higher. So at the end of the day, if you measure the number of responses they had to make for each of those food pellets, it was way elevated relative to air controls. We've also looked at some social behavior. This is our nose-to-nose sniffing, where we've looked at this in relation to a conspecific that's either a complete stranger or is a cage mate. And what you see is that when it's your cage mate, we don't see any decreases in social interaction. But when there is a stranger in this little device here, the amount of contact goes significantly down after these CAPS exposures. There are changes that we also see in testosterone in these animals, which look like they correlated to some extent with the nose-to-nose sniffing. And we're looking, this is not something that we've pursued all that far yet, but it's of interest to us to see how hormonal changes are influencing these effects. So one of the difficulties of doing ambient air pollution is that you never get a direct replication of your exposure. You're pulling air in from outside, so it depends on the day, the season of the year, all of the above. And so what we've done instead is we've done a lot of these exposure studies and just started to build concentration effect curves, looking at some of the same outcome measures across these different air pollution exposures. And here, for example, you can see the ventriculomegaly, which clearly increases with increases in exposure concentration, whereas the white matter is decreasing. We've also seen the changes in glutamate. We see changes in dopamine, which I'm not going to talk about, as well as behavioral impairment. So this is something we continue to fill in. This one is based on all this postnatal data, and it's male specific. So what about gestational exposures? What happens during trimester 1 and 2? We were able to do this study. Our collaborators at NYU, this is Judy Zelikoff, they have a very similar exposure capability, and she was interested in reproductive effects, and we were able to obtain brains from animals from her that had been exposed from gestational day 0.5 to 16.5, and we collected those brains at days 11 to 15 so that we could look at them in comparison to our postnatal exposure. Well, the first thing that you see is, again, you see this ventriculomegaly, but in this case, you see it in both males and females, and you can see that here on the bottom, as opposed to the filtered air controls. So that is seen in both of these exposure periods. We also see some changes in brain structure. At the top, I'm looking at the hippocampus. You see a small but significant, about a 10% reduction in the size of the hippocampus. But unlike what we saw with postnatal exposure, the corpus callosum, that big white matter tract, is now way too big relative to what it should be. And you can see that here. And then we saw this in both the males and the females. And much larger than it should be in terms of relative to control. There was also way too much myelination going on in that corpus callosum. You can see that here at days 11 to 15. These are well over 100% increases, and this was a persistent effect. So even at day 60, you're still seeing a corpus callosum with way too much myelination after these gestational exposures. The ventriculomegaly was persistent in females, but in males, it looked like there was some repair that must have gone on during this process. So one of the things that we looked at was some of the initiating factors in terms of some of these effects, and one of those could be microglial activation. And microglial activation, colonization of the brain with microglia, begins very early. We talk about the microglia in terms of 4 different states. They're actually either very quiet or they could go to being activated. They actually have to be activated for the brain to develop. For neurogenesis and gliogenesis to occur, those microglia need to be activated, but this is a real double-edged sword because an activated microglia is now going to be more susceptible to an inflammatory stimulus like air pollution. What you can see is that in both males and females after this gestational exposure, there is a significant increase in activated microglia that's occurring. So clearly a shift towards a more activated phenotype here. And when we look at the at an ultrastructural level at the myelin, it's not, oops, I'm not sure where we went here. Do we know what? I don't know if I can get back to, do I have to go down here? Where did he? I don't know what. I think Jim left.
Yeah, he left. Let's get rid of this. Get rid of this.
Oh, there we go. Okay. If you look at this, it's not because the axons are wider or larger. It's just because they're wrapping way too much myelin around that axon. And you can see the thickness of the myelin sheath, which was increased again in both males and females after these gestational exposures, whereas there is no change in the diameter of the axon itself. And the myelinating cells of the brain are called oligodendrocytes. And they go through differentiation to become mature oligodendrocytes that then ultimately lead to this myelin formation. And what we saw here again in both males and females was a shift towards a more mature oligodendrocyte, which would be consistent with the fact that there was myelination going on way too early and in excess of what should be happening. So this took us to another sort of way of thinking about this, and will be what I talk about now, which is what causes myelination in the brain. What's essential to myelination in the brain is iron. Without iron, you don't get myelination, or with too much iron, you're going to get too much myelination. So we went and looked at the iron level in the brain. Here you can see the iron content of the corpus callosum was significantly increased after these exposures. And this was particularly the case in females. We didn't see it here in males in the corpus callosum, But when you look at the cerebellum, which is back there by the brainstem where the vagal nerve comes in, you can see that increase in the number of iron inclusions in both males and females, and it's about the same magnitude. So one of the things that really started us thinking about was brain trace element dyshomeostasis as a mechanism of some of the effects that we're seeing. So these are what are called tree maps. These were filters that were taken from the exposure chambers that were then analyzed for trace elements and metals. This is after postnatal exposure, and this is the gestational exposure that was carried out in Tuxedo, New York. You can see this huge sulfur component, no surprise, it's a big component of air pollution, as well as just a whole host of other metals that are on that filter following these exposures, including iron. We see iron in both of these. In addition to that, we took sagittal sections of brains from postnatal exposure and subjected them, this was in collaboration with Brian Jackson at Dartmouth, to ICP-MS and looked at metal analysis. And so here you're looking at filtered air control brains of males, and these were males that were exposed to CAPS. I think you can very easily see the marked increases in both iron and sulfur that are occurring in response to these. Remember, this is a total of 8 days of exposure. They're basically in every region, which is what you'd expect, because both iron and sulfur are required by the brain for development, and they're going to be seen in virtually every region of the brain. They weren't the only things that we saw. These are some of the others. If you look at aluminum here on the top, here on the top, and what's really interesting about that is it just outlines the hippocampus for reasons that I don't yet know what's going on with that. Calcium was also markedly elevated in these brains, and a lot of what we see in neurodegenerative diseases is thought to result from calcium overload in the brain, calcium, as well as copper, marked increases in copper, while at the same time it looks like there's been that these increases have occurred and replaced manganese and zinc in these particular brains. These are data from another collaborator who does high resolution STEM microscopy, and you're looking at 20 nanometer, 50 nanometer sections here where they look at the metals that are in these exposures. Here you can see iron particles, silica particles. Here we have silica, aluminum, and iron aggregating, agglomerating within these particular sections. And there's a lot of work right now to speciate, for example, what iron species is this? Is it something that could be formed endogenously, or is it something that could only have been acquired exogenously? Metals are clearly a component of air pollution. So once again, I'm showing you the 2.5 map here, but here we're looking at the levels of iron, aluminum, silica is a huge problem, sulfur, zinc, lead, and copper. So they are well-known components of air pollution. What they raise is some interesting ideas which have been talked about in relation to neurodegenerative diseases, but not so much with respect to neurodevelopmental disorders. And one of those is a mechanism of action called ferroptosis, which is a caspase independent cell death mechanism that's caused by the accumulation of redox active iron and lipid peroxidation. And that's something that we're in the process of looking at right now. At the same time, as a neuroscience person, you don't really think so much about sulfur and the brain, but there's a transsulfuration pathway, not only in the mouse brain, but in humans, and it's responsible for the production, obviously, of glutathione, but in the process of that is also involved in controlling levels of glutamate, so it could underlie things like excitatory, inhibitory imbalance, and lack of glutathione protection. So one of the things that we've started doing now is generating iron oxide nanoparticles and doing the same exposures. These were postnatal exposures that we've done previously with the ultrafine particles. And what I'm going to show you today, I've done this in conjunction with sulfur dioxide, simply because of data from many years ago saying that the sulfated metals are more problematic in terms of their toxicity. So our hope was to generate some of those. Like air pollution, like ultrafine particles, it looks like iron alone can produce this kind of ventriculomegaly. So one of our questions has been, if we just look at iron or some other metal, how much does it reproduce the features that we see from ultrafine particle exposure? And so, of course, the ventriculomegaly has been a big issue. It does seem to reproduce that. These are all data from postnatal day 14. So again, 24 hours after these exposures end. We also see that it increases myelination in both sexes. And interestingly, it only did it in the cerebellum. And it happened to both males and females. And the reason that's interesting is because if you remember in the gestational exposure, where we had the high iron levels, we saw way too much elevation. We saw it in both sexes, in particular in the cerebellum. So it could be that iron plays a role in that effect as well. We also see that when we do iron dioxide exposures, we get increases in cell death. This is measured by fluorojade or caspase staining. This is in, again, the nucleus accumbens, where we saw cell death in response to ultrafine particle exposure. Here, what's interesting is that we're only seeing this in the females for reasons that we haven't yet completely discerned. Myelination was, again, also increased only in the cerebellum. Well, actually, I think this slide got in there twice. Okay. That loss of neurons that we saw postnatal day 14 in females, what we see is that the trajectory of neuronal development is also blunted in these females, so that by 90 days of age, it looks like it's actually decreasing instead of increasing. There were increases in serum cytokine levels, multiple different cytokines. Again, these were female specific in response to the iron sulfur, and increases in serum oxidized glutathione, which again, we also saw only in the females in this case. There were huge differences in brain neurochemistry in response to these exposures. And what's really interesting is that they're almost always in the opposite direction by sex. So if we just look at levels of glutamate, which I talked about with postnatal exposure as being elevated across the lifetime, you can see that in females, there were actually marked decreases in brain glutamate in these animals. The glutamate GABA, the excitatory inhibitory ratio, increased in males, but not in females. In male brain, catecholamines are markedly increased. This is dopamine and DOPAC and HVA, whereas they were decreased in the brains of females. And again, this is at postnatal day 14. With respect to serotonergic neurotransmitters, the story was a little bit different, but it looks like we have altered serotonin to GABA and glutamate ratios that we see in the females, but again, not in the males. So very marked differences in brain neurochemistry. Increases in astrocyte activation, trying to look at mechanisms by which this occurs. We see those in particular in the males. It looks like the male astrocytes, for whatever reason, are more susceptible to this iron. So significant increases in astrocyte activation in males in olfactory bulb and cerebellum, as well as in nucleus accumbens, but only in the cerebellum in females. And again, we know very little about differences, sex differences right now in terms of astrocyte function. And I'm going to end by talking a little bit about one of the directions we're also planning to go on in the future. One of the things that we looked at in these brains after the iron exposure were levels of beta-amyloid and tau. And the reason for doing this was because every neurodegenerative disease I'm aware of has a marked elevation of iron in the brain, and no one's ever been able to figure out where that iron is coming from. Is it just that your iron metabolism is screwed up? Nobody's stopped to think about the fact that you're breathing this stuff in your entire life. The brain doesn't appear to have a mechanism to eliminate excess iron. It controls the entry of iron at the level of the blood-brain barrier. So it probably never needed a mechanism to get rid of too much iron. So it has a very, very long half-life. And could that be a fetal basis? Could something like early or lifetime exposure to things like iron and air pollution be a basis of later neurodegenerative disease phenotypes? And so we looked at levels of beta-amyloid and tau, and I think you can see here they were elevated, significantly elevated in females. This appears to be a lifelong effect. It was still being seen at day 357, whereas you don't see any of this in the male brain. So in summary, I hope that I've convinced you that exposures to real-time, real-world, human relevant levels of ultrafine particles during periods of brain development can produce very enduring characteristics that have features in common with multiple different neurodevelopmental disorders. Obviously, and of no surprise, the effects are going to differ by sex because brain development differs by sex from the first cell on. And with the developmental period of exposure. One of the things that we know about many neurodevelopmental disorders is there is just a huge range in terms of their phenotypic and behavioral expression all over the map. And one of the interesting things about thinking about air pollution as a contributing factor is that so is your exposure to air pollution. You could have the same mothers in the same town, but living in different parts of town being exposed to different components, levels of air pollution on the same day or different days. So there's a correspondence in terms of the variability of these kinds of exposures in some ways to the phenotypic expression. I think it's critical to start asking about the components of air pollution that might be responsible for these effects. One of the things that can do is allow us to build better animal models so we can look more specifically at mechanism and have the other thing that we could then do is think about intervention strategies, but I think it's also really important to public health protection. We regulate levels of PM10, PM2.5. There has been discussion, which hopefully will pick back up now that we've changed administrations, about trying to regulate levels of ultrafine particles, which is no small problem. But we do regulate levels of gases in the air, and we regulate levels of lead in the air. There isn't any reason why we couldn't have regulatory levels for things like iron and or copper and silica that are huge problems. So a lot of work to do, a lot of opportunity. And I just want to thank all the folks who have been involved in these studies, and Günter Oberdorster for dragging me into this, kind of kicking and screaming at the beginning, but I've learned an enormous amount, and we couldn't do these studies without him. My collaborator, Ushi Graham at the University of Kentucky, who does the high-resolution scanning electron microscopy, and Brian Jackson, who is a member of the Superfund at Dartmouth, as well as all of the members in my laboratory who've been involved. So thank you very much, and I'm happy to try to address any questions that you have.
So we'll take questions from the live audience first, and Sarah is monitoring the chat. So those of you who are online, if you have questions, raise your hand, and we'll call on you as we can. Cindy.
So let's talk about those sex differences.
So this, I think at the postnatal period, one of the things that has been looked at is microglial colonization of the brain. So we know that at that period, there is a greater colonization of the male brain with activated microglia. That switches over then by like postnatal day 18 or 20, where it goes up in the female. And as I said before, those microglia have to be activated for neurogenesis and gliogenesis to occur. If you screw that up, you change the profile of cytokines, you stop neurogenesis. And so one of the things we're planning to do is to do exposures a little bit later when that window shifts and see whether females would be more vulnerable at that point and males less vulnerable, or maybe show a different kind of phenotype. During gestation, it's very unclear. There's lots of data following the day of birth in mice on in terms of brain development, very little in terms of gestation. And I mean, it's not a trivial question to try to address experimentally. So I don't know what's going on during gestation that could account for the kinds of sex differences. We don't know whether are females just more vulnerable to iron in particular. We don't know that.
You can manipulate hormone levels. One thing different is testosterone.
Yep, yes, and something we're also very interested in doing, yeah.
Alex?
Has anybody looked at like the percentages of ultrafine particles free versus the bound, the PM 2.5 versus the PM 10? And then secondarily, kind of building on that question, when we go indoors with air conditioning stuff, do we get reprieve from maybe the ultrafine particles, or is that information?
The answer is no, you don't get reprieve, unfortunately. So in fact, it's easier entry for them in some ways, certainly than the larger particles. With respect to the percentage, I don't know the answer to that question. I mean, there's a whole group of people that does nothing but those kinds of studies, and I don't know that I've ever seen anything with respect to what those percentages might be.
Derek?
Do you have any ideas for some kind of therapeutic strategy to get iron out of the brain?
Well, there's a lot of people that think they're going to chelate their way through this, which is just a horrible idea. But you'll see this in the neurodegenerative literature all over the place. That's not going to work. The one thing I do think about, there's a lot of work right now going on the glymphatic system of the brain and the potential, maybe the potential to eliminate iron somehow at that point. But I don't think we know enough about that yet to know that that's possible. There is transferrin there and some other iron binding proteins, apparently though in very limited quantities.
Rachel?
So for your collection of ultrafine particles from exterior air and concentrated, do you see a seasonality effect or impact on your phenotype of the mouse model?
We haven't done it that way. So I haven't ever separated it that way. I tend to try to do those exposures mostly during the cold season because that's when you have higher levels of this. We haven't tried to do it that way. There's clearly going to be differences every time you do one of these exposures, whether it's in terms of the metal profile or the organics that are adsorbed. There are always going to be differences. So in some ways you can never do, apples to apples in these kinds of experiments.
Do you think these effects of this exposure are reversible? Like what happens when you take a CAPS exposure mouse and put it back in the chamber? Does this effect go away? Are you reversible, not reversible at all?
Wait, say that again. I'm not sure I was following you.
So the effects of the exposure are these reversible? Like let's say you have exposed a mouse and then put it back in the filter.
I would have to do it systematically. My sense is that that if the damage exceeds a certain point, I don't think there's any way to repair it. And some of the magnitude of some of the ventriculomegaly that we do see a real range of it, but some of that is probably never reversible. It's just too much. What would happen if I did it on a 1-day exposure, 2-day, 3 would be interesting. We haven't done anything like that as of yet to see. Or if we stopped at postnatal day 7, clearly we would have less, but that's not what happens in real life. So
I was really interested in your data from your collaborator with the University of Kentucky. It looked like that, and maybe I'm misinterpreting the colors on your slides. It looked like that the metals were actually being deposited in different types of, or different cells at least. Is that correct?
I'm sure that's true. There's also going to be differences in the shapes of those particles depending on their speciation. So yeah, that could very well be the case.
Yeah, I was just wondering if that could explain some of the differences between sexes in terms of where the metal goes in terms
Yeah, and one of the things that we want to do is to try to track that, trying to maybe use an isotope or something and kind of get a sense of where things are going. But this helps too because she can speciate that iron and say, this couldn't have been, this has to be exogenous. It couldn't be something that was formed, you know, in vivo kind of thing. So.
Yeah, so I was kind of curious when you brought up the idea of the spaggy mouse, and I didn't realize that the postnatal day, whatever, 7 to 14, I don't have that range, and the mouse is the equal to the 3rd trimester in humans. So in your model, I'm thinking about, so they had an inhalation exposure.
Correct.
But that's not what's going to happen
Correct.
in humans.
Which is why I'm so interested.
Mice,
Yeah.
Do you think it would be different?
Yeah, because they basically are born, so they're an African species of mouse, and they have a 39-day gestation period. So a normal mouse is 21 days, right? They come out, they're like good to go. So that's one of the things that we're going to do is do the exposures in these spiny mice and see what the difference is. What is the contribution of that inhalation during that period? So I have a colony going. The downside of spiny mice is their litter size is like about 2. And they're kind of like voles in that they're very bound socially. So when you start separating them, you start getting issues as well. But we are building up the colony, so we're going to start doing some of that.
I wonder, really quickly, if you are, the things you're seeing different in the 1st and the 2nd trimester in the mouse versus the 3rd, if those will actually carry over because it's the route of exposure, the route's going to be different. Correct. And I think I can get a better separation in a spiny mouse of 1st, 2nd, and 3rd trimester. That's been pretty well worked out, interestingly enough.
Yep.
So the brain gets rid of toxic stuff by using the lymphatic system. How come the brain doesn't get rid of all the metals and those ultrafine?
I think you have to go back literally to natural selection. The brain didn't need a reason to get rid of excess metals. There weren't situations causing excess metals really until about the time of the Industrial Revolution and then when air pollution picked up. So the brain didn't really need to have something like that. It was so controlled in terms of its uptake into the brain at the blood-brain barrier that there wasn't a situation where you had excess.
This crosses the blood-brain barrier, right? And it accumulates as soon as something accumulates.
It's not crossing the blood-brain. The problem is it isn't crossing the blood-brain barrier. It's literally going directly up the nose into the brain and bypassing the blood-brain barrier. So unfortunately, none of those mechanisms are operative. You're going directly up the olfactory axons, the trigeminal axons into the brain. You're bypassing all that regulation. And in a fetus, a human fetus, that iron regulation it's not really fully in place, I think, until about 6 months of age. So even after a child is born, you've still got 6 months where you could, depending on where you are, be breathing that up. But I think it's mostly the brain just didn't need to.
Rachel, number 2.
So could you speak a little bit about establishing the spiny mouse colony and whether you plan on looking at reproductive health of the spiny mouse colony with the reticulates? And the reason I'm interested is because the spiny mouse has a menstrual cycle.
Yeah, we will be, it's a slow process of developing this colony. And we don't want to inbreed them, so we're trying to spread them out in terms of building the colony. But yeah, we will do gestational exposures and we'll be able to look at things like that as well.
Where are you sourcing your spiny mouse?
They came from somebody who has a big colony. I think it was Ithaca. I'm trying to remember, but he had a very large colony, so he sent us 8 breeding pairs to get them started. Somebody else had gotten them from 2 breeding pairs from a pet store, but that just didn't quite seem like it was going to work. So,
Kirk, very nice talk. A couple of years ago, my lab reported that there's a very big difference in male and female mice with respect to their circulating levels of glutathione, which we talked about. And interestingly, there's also a very big difference in the amount of cysteine in plasma between the male and female mice. And you didn't really talk about it, but the the link between your glutamatergic signaling and ferroptosis is the same cystine transfer.
Yeah, I'm very interested in that. In fact, we have a proteomics core, and I just asked them whether they could take some of our tissue that we've already processed for other things and run that whole panel. Because one of the things I'm struggling with is what happens to a brain that is seeing too much sulfur? And it's really hard to find any information on that. But so one of the things I want to do is look at parts of that pathway just to see if we can get a sense of what might be going on. And there's some very interesting, excess cysteine, homocysteine are real problematic for the brain. So is that a part of this? Yeah, I agree. That's really interesting.
Thank you so much for coming and giving this talk. I was really interested in your slide about the different kind of relative concentrations of metals that you're finding when you take those filters. I guess I have two questions. Are those filters catching PM 2.5 and PM 10 as well?
No, because they're not. So those were done. They're different from the ICP-MS that was done on the sagittal brain sections, but at the level of filters, that is concentrated ultrafine particles. So we actually separate out larger particle sizes from the delivery to the chamber. So no, you're not getting that.
And then my second question, do you think it would be worth repeating these kinds of experiments in different areas of the country to see?
Oh, absolutely. Absolutely. Yeah, absolutely. The problem is there are very, very, very few places that can do ambient inhalation exposures. And so one of the things we're starting to see is a lot of intranasal instillation or tracheal instillation as sort of another mechanism to consider air pollution kinds of things. There's so much chemistry that goes on in air pollution that you are missing by doing something like that. So they're basically scraping particles off filters and then going through a process where they resuspend them, or just taking those particles and putting them directly in the nose with the thought that they're going to go directly into the brain. But that is a very different exposure than ambient air pollution. So I'm not sure how informative it is.
Okay, before we move to any questions in the chat, I want to make sure that we thank the CIEHS office staff, Colleen, Ariel, and Sarah, who made this possible by making all the arrangements. Sarah, is there anything in the chat?
Yes, we have lots of questions in the chat. First question is from Dr. Kai. How about body weight, brain size / weight, and the cortex development when pups exposed to concentrated polluted air? And is there a mouse strain difference in response to exposure?
I guess I should I, do I need to talk into there? No, I just don't know if I need to talk into it. Okay. So there are no body weight effects of these exposures. We have not done brain volumes. We actually have a collaborator at Duke who is going to be doing that for us. He does 3D MRI in rodent brain, so he will be looking at that, but we haven't really seen anything there. What was the second part of that question? It was different strain. I would be surprised if there wasn't a mouse strain difference. I'm sure there would be, but we haven't looked at that specifically.
And then we have a second question from Dr. Natalie Ducray. She says, this was an excellent talk. Thank you. Do you all see corpora, amylacia, in these brain tissue specimens exposed to high air pollution? I know they are common to see for those with neurological disorders like Alzheimer's, also prostate cancer, and the corpora amylacia are rich in calcium, inflammatory proteins. They are thought to be indicative of past harmful exposures. I wonder if they are involved in here with air pollution.
Interesting question. We haven't looked. Haven't done a lot of that pathology. A lot of our initial focus was more white matter stuff, but it's something that would be really interesting to look at. And we save all the tissue from all these mice, so if she's interested in looking at any tissue, happy to send it.
Thank you.
And Dr. Alex Carl said, great presentation as well. Can you address what size PM can make its way up to the olfactory and trigeminal nerves.
Trigeminal, yeah.
Quite some time ago, Auburn Dorster demonstrated about 20 nm gold particles made it to the brain. But are you observing much larger UFP are translocating to the brain also?
Yeah, our particle size for the ultrafine particles ranges from about, well, up to about 100. We know that those are going up there. I don't know that anybody's looked at the largest size particles. I do know that the smaller the particle, the larger the uptake that you will get into through the olfactory, up the olfactory axons. So it hasn't been all that systematically carried out yet. He did those experiments many years ago, started with the gold nanoparticles of about 20. Our iron particles are around 20 nanometers in diameter.
Thank you.
Then we have another question from Dr. John Wise, Jr. Did you measure any other metals with the ICP-MS analysis in tissue? Most of the metals you shared were developed metals. So I wonder if the developmental transporters are hyperactivated or expression has increased. Or if other metals are accumulating, I wonder if that may reflect BBB damage or leakage.
Yeah, interesting question. So when we did the ICP-MS, we could select some 12 metals that we could look at, and there clearly are a host of others that could be looked at as well that we simply haven't done, but certainly could be done. And we have tissue sections remaining to do that, and we'll be continuing to look at that. We're going to extend the number of metals that we look at or run multiple sections with different metals.
And then we have one more question from Dr. Vicki Hines-Martin. She says, my focus in our center is serving as a bridge between our communities and our science. So one clear responsibility is to translate our science into language that communities can digest and see the relevance or future relevance to their lives. So what messaging do you use? This is of particular importance when you look at COVID vaccine hesitancy because communities that the development was too quick, or perhaps this could be avoided with info to the public.
I don't know what the actual message would be. I mean, this is where we always get into let's not scare the public or blame the mother kinds of mentality. I do hope that as we understand more about the influence of air pollution on the brain, if it is contributing to neurodevelopmental disorders and or neurodegenerative diseases, that there is a groundswell of people saying we got to clean the air. Cleaning up, going after the ultrafine particles is not trivial. I can be in a block in New York City, and one end of the block, you know, there'll be a spike in ultrafine, and then be 20 yards away, and there's nothing. So it's not going to be an easy task to do. So I don't know what we tell people at this point in time. Not sure I have any answers for that yet.
We have a hand raised. If you unmute yourself, we should be able to hear your question, Effie.
Okay, hi, my name is Afi. I'm an R25 student with Dr. Hein and Dr. Hong. And my question was more about the public health implications of this study. Since it's based on demographic and air quality, I think that one thing that would be worth considering is the racial demographic when it comes to quality of air. So my question is more to, is there someone focusing on this, or are the studies along those lines in relation to this particular topic?
So one of the things that is interesting about air pollution is in terms of toxicology, it's probably a more equal opportunity exposure. So think about how cities, parts of cities get rebuilt, wealthier people move into those parts of the city, they're all driving in the city. So in some way, and that's obviously where a lot of the pollution is, in some ways it has become more of an equal exposure than some of the other toxicants that we think about. And I'm not sure if that answered the question or not. Certainly relative to some of the other things I think about, lead or some of the other metals, it's more equally spread to some extent.
There are no other questions or hands raised on the chat.
All right. Well, thank you.
Well, thank you all very much. Oh,
Very
somebody else.
Maybe one point about that. I think the DP will challenge that notion just a little bit because communities of color have more industry and more polluters and less green space in our green heart study.
Yeah.
So I think it'd be good to dig deeper on that.
Yeah, no, I agree.
On the surface, but.
Yeah, right.
And
Agree.
they have less identification of neurodevelopmental disorders.
Okay.
Okay.